Next up, we have Skyler Elmstrom. He will be attending Western Washington University this coming fall, and he's already been taking some math classes over this summer, too. He intends to major in environmental science with an emphasis in freshwater ecology or marine science, and minor in geographic information systems. He's been working here in Newport with the Shellfish and Estuarine Assessment of Coastal Oregon, SECOR, with the Oregon Department of Fish and Wildlife this summer. And one of his ambitious goals this summer was to read an entire book. So, we'll see.
Just going to be a couple minutes to load the application here. Which button is it to pop it up on the screen? Oh, okay. All right, let's go. It's not reading. I'll try.
Oh, nice.
Okay. Technical difficulties. Sorry about that. Hello everyone, again, my name is Skyler Elmstrom. I'm working with the Shellfish and Estuarine Assessment of Coastal Oregon team, or SECOR. They're a permanent project through the Oregon Department of Fish and Wildlife. Our main objective is to assess bay clams and their habitats in all of Oregon's estuaries. For this summer in particular, SECOR received a grant to evaluate small unmanned aircraft systems, or UAS, more commonly called UAVs or drones, as a tool for estuarine management and research. After negotiating the unique learning environment of mud, rising water, and not so waterproof flying robots, the occasional disgruntled seagulls, and many hours of processing, I'd like to present some of our preliminary results to you guys. On screen is one of our mosaics from Netart's Bay, Oregon. A mosaic is a collection of stitched images from our UAV flights. So the big question we're trying to answer here is, what can we see from our imagery? Pretty basic, but very important for us. So ideally, UAS would allow us to more precisely track changes in estuarine environments and measure the extent of the essential habitats that we're looking out for. UAS would require less staff, give us access to otherwise inaccessible or unsafe parts of estuaries, and allow us to increase the frequency of how often we visit each site. Aerial images also increase our sample coverage compared to ground crews, which is otherwise challenging or time-consuming, given that all of our surveys must be conducted at low tide. Initially, we were unsure about what to expect, but our preliminary results show that we can not only identify the presence or absence of our target species, but also count them and possibly measure them as well. So coming up are a few examples that I've selected to show you. This first example, a very dense Tresis capex bed, or gaper clams, as they're commonly referred to. Throughout our sample area, we place ground truth quadrats that are 1 meter squared to help us determine the reliability of our images. So an example here from our mosaic and our photo that was taken by the ground crew. Orient ourselves over here. Each quadrat has one corner that's painted red. I've highlighted that in red here. And there's another feature in the picture that may be hard to see in the back, but it's a hinge of a gaper clamshell. So once we orient ourselves, we can begin to count, and it's very clear in both images that we can actually count gaper shells, which are about a quarter in size or larger. So just starting counting here, 1, 2, 3, and so on. Oh, sorry. If we zoom in even further, we can see they're siphons. This was a joke. we can't actually zoom in this far, but we did get a pretty good aerial image of these siphons submerged underwater from our UAVs. So I've selected another photo. It may not be visible with the lighting conditions in here, but there are quite a few that are protruding from the sediment under the water, which was impressive to us. We didn't expect to find anything underwater. So I've highlighted them in purple for gaper clams. There's a small group here another one here and there. And upon closer observation of these images, we could also see active pumping from the clams. We could see ripples in a bullseye pattern around from where we could see them. In my next example, I have the butter clams, or the Saxidomus gigantea. These holes are much smaller. We were not sure at all if we were going to be able to see these compared to gapers, which gapers usually are at least a few centimeters. Butter holes are keyhole shaped or jelly bean shaped, much smaller, about a centimeter. We were actually able to observe these too. Highlighted yellow, there's a small butter clam bed and some other sites around that one as well. In my final example from the mosaic, I chose a site that had both butters, gapers, and the vegetation that we're looking for as well, eelgrass and macroalgae. Go through here really quick. So again, the butters in yellow, the gapers in purple, several locations. Macroalgae, that was easy to differentiate from our eelgrass. And then our eelgrass highlighted in the lime green here. Not pictured here, we were also able to distinguish the invasive eelgrass, the Zostra japonica. So what's next for our project? We haven't really dug into the data yet. We haven't really calculated, quantified the amount of features that we've collected with our imagery. So that's next in line for us. But ultimately, we hope this information will help us determine habitat boundaries in particular, help assess changes in those estuaries over time due to climate change or other factors, assist with evaluating agriculture, aquaculture, permit leasing, and the impacts that those leases could have on these habitats. Additionally, these large data sets will be available permanently for other researchers and resource managers. So that concludes my presentation. I'd like to give special thanks to Oregon Sea Grant, the Secret team, and my fellow summer scholars, especially my team. All right, thank you. In the back.
Can you talk a little bit about the regulatory challenges of flying drones over areas like this?
Oh yes, so the regulatory environment of UAVs, as some people kind of know, is very, very confusing, especially for commercial versus agency flights. So for the Oregon Department of Fish and Wildlife to fly, we actually have a COA that's been approved by the FAA to allow us to fly in certain areas under certain conditions. So doing that, we have to log our flights, we have to communicate with air traffic controllers about where we're flying, when we're flying, how high, for how long. They send that out to other pilots, and then they give us permission to fly. In the future, actually later this month, they're releasing new guidance on and regulations on how to go about flying UAVs. It's going to be much more streamlined. We can submit applications online to the flight traffic controllers. Much faster process, much less burden on them to approve us for varying flights.
Thanks.
No problem. Go ahead.
How do you determine what area you have in your photos?
So the areas in our photos are actually sampling polygons that we've selected previously for other habitat collections. Those are all based off of either sites that we've visited and determined would be good representative sites for the information we're trying to collect.
Well, I mean, how do you know if it's half a square meter or
Oh.
a meter?
Okay, so part of what we did, we conducted ground truth measurements. So we would take a RTK GPS out with us and collect solid ground points. And then with that imagery collected, after we stitched it all together, we would use those points to give some kind of geospatial reference to our image. And then from that, we could do measurements. Go ahead.
How'd you go about, could you speak to something about the accuracy and the completeness of your characterization of the shellfish
Sure.
species?
So are you talking about the accuracy of our mosaics?
Yeah.
Okay, so, gotcha. So the imagery itself, without that GPS RTK, it would have been accurate to within 2 meters of the actual position on the ground. The pixel resolution of our image ideally a sub-centimeter, but for most of our images, we got below 2 centimeters.
I was more like, how do you know that you really have that species of clam? And
Right.
did you get them all in an area?
So we're still trying to hash that out right now. So with all of our preliminary data, we're just really trying to like, hey, can we see these things? All right, so if we can see things, let's try and figure out a way to make sure we're identifying them correctly. And then hopefully we'll be able to use software to help us do that automatically so there's less error with us doing it manually. Go ahead.
Really impressive, nice job.
Thank you.
I'm wondering if you have any idea about how much time and effort there is in processing all those data
Oh,
that you're collecting, and how that compares to actually walking out and looking up this.
okay. Well, I'm your man for the processing. So, I actually acquired a nickname, Screen Burn, because a lot of my time was looking at loading bars and waiting for processes to complete. So for the area that we did, I want to say it was roughly about a square kilometer or 2, depending on where we were. And processing that, depending on how you want to do it, there's various settings that you could do, but for just like a rapid assessment, it's probably like 3 to 4 hours of processing. If you want to do something that's far more accurate that we'd probably use for measuring, it's going to be 7+ hours depending on the data set. So there's a lot of processing involved. Compared to those field surveys, it can't even compare, because to analyze that amount of space on the ground that these UAVs are taking with aerial coverage would take a lot more hours than we have to work, especially with those low tides. Okay, is there anybody else?
Cool, thank you.